The goal of this protocol is to describe changes in motor behaviors in individuals with incomplete spinal cord injury following administration of serotonergic medications. To accomplish this, standardized assessments are performed to evaluate clinical measures of baseline lower extremity strength and reflex behaviors. To obtain more precise measures, quantitative static assessments of strength and reflex function of selected muscle groups are determined, with specific investigation of peak volitional torques and stretch reflex excitability. Additional quantitative dynamic assessments are then performed to measure lower extremity kinematic patterns, muscle activity and timing, and metabolic efficiency during treadmill ambulation. Finally, serotonergic medications are administered in a blinded crossover design and assessments are repeated. This approach of translational medicine systematically assesses the effects of serotonergic medications on volitional strength, reflexes, and walking ability in individuals with incomplete spinal cord injury.
Spinal cord injury is a debilitating disease process which produces profound deficits in motor control. Early after injury, a primary goal for most patients is the recovery of walking. In individuals with an incomplete lesion, meaning they still have residual motor control distal to the site of the lesion, this goal may be obtainable. Animal models have shown that pharmacological agents can help accelerate and promote the recovery of motor control and walking ability. However, the translation of these findings into humans is limited. Our lab is trying to translate these animal findings into humans with incomplete spinal cord injury. We hope to ask